Morning, everyone. Welcome to the Arthritis Web Seminar Series. Today is our lecture number 34, and it's going to be on gut microbiome and arthritis. Our guest speaker today with us is Dr. Jose Scher from New York. Welcome, Jose. Before we start the introductions, just a reminder to the audience, please post your questions on the question answer tab on Zoom. Rob?
Good morning, everyone. It's a great pleasure to welcome Jose Scher to our web seminar series today, virtually. Hopefully it'll be live at some point. Jose is an associate professor of medicine and director of the Psoriatic Arthritis Center at New York University in New York City. He received his MD in Buenos Aires in Argentina, and then completed his internal medicine and residency at NYU, where he's now on faculty. His research interests have included immune and environmental factors that enhance the transition from psoriasis to PSA, the mechanisms behind the microbiome perturbations in psoriatic disease and rheumatoid arthritis, and particularly the pharmacomicrobiomics of DMARDs and immunotherapies of RA and PSA. And Jose has established an outstanding leadership profile in this complex and changing landscape of the microbiome, and has done an outstanding job leading the PSA agenda in North America and Europe, and it reports that actually that the PSA program there is now under the AMP umbrella from the NIH, which is very exciting. Jose, welcome, and great having you here today. Thank you for joining us.
Oh, thank you so much. Thanks, Rob. Thanks, Rohit, Mariam, as well, for organizing. So I'm told that I have 35 minutes, and I intend to keep it on time, except I always promise to do that, and I never deliver. But thanks for all of you for being in the audience. I'm going to talk about microbiome in general initially, and then what's occupying our minds more recently, which is this pharmacomicrobiomics story. So these are our funding and conflicts of interest, and a little bit of an outline. Again, I'm just going to go briefly about what has happened in terms of the potential for microbes, particularly microbiome, as triggers for immune-mediated inflammation, excuse me, and then delve into pharmacomicrobiomics, initially of immunotherapies, a little bit about the cancer literature, which actually led the field, and then talk about some of the biologic stories, and then DMARD's dependency on gut microbiome. Can microbes trigger psoriatic disease in particular, or inflammatory arthritis of any kind? And the answer has been, for the most part, yes. You know, there's classic examples in the human environment that pertains to things like strep infections triggering gut-age psoriasis, for example, tonsillectomy, anecdotally at least, leading to amelioration of psoriatic disease as classic examples. Obviously, reactive arthritis being the classic example on how an infection of the intestine can lead to peripheral arthritis. But what do we know from animal models? And this slide is going to be defective because I just tried it, but it doesn't really matter. I think this is true in most animal models, where when you rear these animals under germ-free conditions, these animals, for the most part, do not develop disease. This is an example of SKG, where the germ-free animals are free of the phenotype. If you now raise them under conventional circumstances in regular cages, you start seeing synovitis, there's psoriasis, and there's dactylitis. You can see the histologic correlate here. And, you know, this is just one example in that there is an IL-23 dependency. This is a Cortland-dependent model where fungal cell wall patterns can trigger disease. But if you treat these animals with a P19 blocker, the disease is ameliorated or prevented. This is true for the HLA-B27 transgenic rat. It is true for the collagen-induced arthritis model, and on and on and on. So proof of principle that that is indeed the fact. It's not any microbes. If you utilize under experimental conditions certain bacterial cocktails on the animals, you will recapitulate the phenotype, but that's not true if you utilize different microbial communities. In the human domain, this has been true in every disease so far. Yes, this is correlative in nature, but multiple groups have shown that a process called dysbiosis, this alteration or perturbation in the homeostatic process of the microbial communities, particularly in the gut, can be found in every autoimmune and immune-mediated rheumatic disease. You know, whether we're talking about psoriasis, PSA, rheumatoid, or AS, this has been true and true. And of course, the biggest challenge has been what to do about it, and whether there are any mechanistic implications for this dysbiotic process. This is some of our newer data, because the idea is that not only we are finding a dysbiotic process in the gut, but if you look at Julia Manason's work in the lesional and non-lesional skin in psoriasis, PSA, and in healthy individuals, what you find is that when you apply several computational metrics to characterize degree of similarity among these three networks, you can see the healthy network, the lesional skin, and the non-lesional skin. What you're finding is that the non-lesional microbial signatures from psoriatic participants are closer to the psoriatic plaque than to the healthy skin networks, which is to a degree not unexpected, right? We've known this from histology, that the non-lesional skin is actually not healthy, because there is a process of a preclinical state, if you will, suggesting one potential mechanism to explain, for example, Kepner phenomenon, right, in this disorder. Why is it? Do you have a predisposition to develop psoriasis in a clinically non-evident skin? And then on panel B, what Julia is finding also is that non-lesional skin here in figure B, there's psoriasis-specific taxa, and I'm not going to go into names, but Acinetobacter being the more relevant one. They're significantly decreased or absent from PSA, which, you know, in turn, this can be a potential help, particularly as we're trying to find biomarkers for disease progression from psoriasis to PSA. Okay, we've done this work many years ago, this idea that gut dysbiosis is also true in psoriatic arthritis. And not only that, but it resembles what we find in IBD patients. This is the fact here. If you look at phyllodiversity, both PSA and psoriasis patients have a significantly decreased phyllodiversity compared to healthy individuals. And it's not any microorganisms, but rather those that are beneficial commensals, particularly Clostridia, Rhinococcus, and others that are either decreased or absent in PSA, and also true, found to be true in ulcerative colitis and Crohn's disease, which is not, again, unexpected, because as we all know, and I don't have to explain to the audience, that PSA is part of the spondyloarthritis umbrella, and therefore it follows that you will find some commonalities in between IBD, axial SpA, and peripheral SpA as well. So it may not be related to the microbes per se. There's a lot of work being performed and pursued in metabolomics, particularly in the active short chain fatty acids, particularly when it comes to expansion and maintenance of regulation of certain regulatory T cells in the gut, and prevention of downstream events. As you know, dietary fibers are not digestible unless you have certain gut microbes. And if you do, those dietary fibers are metabolized into short-chain fatty acids. The most relevant ones to date are acetate, butyrate, and propionate. And those are not inert because they have biologic properties in that, particularly true that regulatory T cells have certain receptors, GPR46 and others, that now are useful to activate those cells, and in turn, the ability to suppress pro-inflammatory downstream Th17 cell activity, in this particular case, several years ago, shown to prevent an IBD in an animal model. And so what happens in inflammatory arthritis, right? Is that only true for local inflammatory process? The answer is no. It's also true in rheumatic diseases, particularly collagen-induced arthritis. What you see here is that both acetate and butyrate actually prevent the emergence of a arthritis phenotype in a collagen-induced arthritis. And you can see that over time, after immunization, you can see the pathological score being decreased with these two short-chain fatty acids. And here you see the histologic correlate, where acetate and butyrate appear to decrease the inflammatory process in these particular animal models. Not only true for CIA, it's also true for the KB/N serum transfer model, in that if you now look at short-chain fatty acids, they can ameliorate over time the total arthritis score. And importantly, what you see here is the fact that introduction of a high fiber diet can also emulate the activity of short-chain fatty acids, which is reminiscent of this graphic here. You can decrease the total arthritis score over time just by introducing a dietary change. So, okay, that's all fun, and that's animal work. We can cure mice. So what? Is it possible to modulate the intestinal microbiome? In any kind of human inflammatory mediated disease, arthritis in particular, PSA and others, we've known for now about a decade that you can do this through various means, all the way from altering the entire ecosystem, such as in fecal transplants, where you modify hundreds of strains, but truly don't have an understanding of the totality of the composition of the microbiome. And you can go into consortia. Examples are lactate producers, for example. You can go into single strains, one strain with a pure isolate. And most recently, the idea that you can utilize bioactive molecules that are produced by certain strains and ultimately mediate effects on the host. So that's in the making. There's several companies and startups looking into these possibilities. What happened in the humans, this is work by Georg Schett's group, recently published the idea that indeed high fiber diet can increase circulating short-chain fatty acids, and at the same time can lower pro-arthritogenic cytokines. This is true 30 days after this high fiber diet in in rheumatoid patients, where you can see that not only the total, but the individual short-chain fatty acids go up, acetate, propionate, butyrate, and some of the microbiome perturbations. And they correlate systemically with certain pro-arthritogenic cytokines, including MCP1, IL-18, and IL-33, all of which decrease after a month of high fiber diet. So the role of high fiber diet in arthritis needs obviously further investigations. This data does not include clinical amelioration. It may be a short period of time, relatively speaking, but that's where the field is going. For those of you who are going to ask about dietary changes, this is one element to consider, that there may be some truth to the systemic inflammatory modification. But let's go all the way up into the fecal microbial transplantation as a strategy being utilized to modulate the microbiome in disease. Of course, it's administration of fecal solution from a donor into an intestinal tract. There are different routes of administration, upper endoscopy, colonoscopy, enema, and most recently frozen cups. Several companies are going through this process of freezing material so that they can be delivered into the right place. The indications currently, of course, FDA approved for refractory C. diff infection and experimentally in IBD, and I'll tell you a little bit about their work in PSA. Okay, this is just one example published in Lancet 4 or 5 years ago now, multi-donor intensive fecal microbial transplantation for active ulcerative colitis in a randomized placebo-controlled fashion. This is what they find, is that FMT is indeed superior or was superior, excuse me, to placebo, 27% versus 8% in achieving both clinical and endoscopic remission in ulcerative colitis. Curiously and importantly, the secondary outcomes achieve similar numbers compared to the biologic therapies that we see in clinical trials, which is suggestive of the fact that if you perform a multi-donor intensive FMT approach, then that may be enough to modify the trajectory of local inflammatory bowel disease. And emphasis on intensive because this was performed daily for multiple weeks, and therefore has to do most likely to the colonization process and other not only donor but recipient aspects of being able and capable of receiving foreign material. So more on that, hopefully soon. What happens in PSA? This is the anecdotal evidence published a year or so ago. A patient who underwent fecal microbial aorta transplantation for a different indication, in this case C. diff infection, resulted in a decrease in PSA disease activity, which was deemed to be drug-free disease remission. So this is anecdotal what happened most recently. So this is the group in Denmark, which I'm sure you read this one a couple months ago at the Annals of Rheumatic Disease, the publication of the results of the safety and efficacy of FMT for active peripheral PSA, and then they had to include this word, exploratory, and I'll tell you why, randomized placebo-controlled trial. A couple of things with this study, it was significantly underpowered. Their power calculation was north of 80 patients, 40 in each group. They ended up enrolling 15 patients in each group, and therefore that's why ended up being exploratory. Second is a primary endpoint, is an unusual one. It's a composite outcome of the proportion of participants with treatment failure at week 26. And that was considered through a shared decision-making and based on the need for more than one intraarticular steroid injection or the new prescription for either conventional synthetic or biologic TMOR. Reminder that these were patients with background methotrexate that underwent one and only one FMT procedure. So that's the other issue with this trial, but these are the results. So curious in that the sham group, meaning the placebo group, did significantly better than the FMT group at week 26. If you look at this, 60% of the patients that received the FMT failed the approach in that they required mostly biologic treatment, compared to only 19 of the patients receiving the sham solution. Curiously, the ACR 20s, 50s, and 70s outcomes were fairly robust for the sham solution. I don't know that we see ACR 50s and 70s with any biologic treatment. Certainly there's no placebo control, true placebo to the sham solution, because we don't necessarily know what the sham contains. But lots to discuss here. You know, there's the lack of power, but also the need for microbial features that are not part of this study. We don't know what kind of donors were used in terms of FMT. We don't know how much of the fecal transplantation actually engrafted in the recipient, and therefore it's hard to interpret except to say that this is the most relevant one. I'd say that it may very well be, and this has been commented on by others, that this is not that the sham group did better, but rather the FMT group did worse in a reactive arthritis fashion. Why is it that 60% of these patients at day 30 on average had worsening of the disease? It may very well be that the modulation was the opposite of what was expected. Lots and lots to do and learn. There's the 2.0 for this study, and hopefully we can incorporate other features, most notably the microbial and hopefully metabolomic features, to see you know, what's underlying these differences. Okay. Pharmacomicrobiomics, I'll start with immunotherapies and then jump into biologics. What are the clinical implications? These are not irrelevant in that we know that the human gut microbes can metabolize xenobiotics. It is true, obviously, for dietary compounds, some chemicals as well, but most relevant to what we're trying to achieve is the idea that the gut microbiome can chemically modify pharmaceuticals, the ones that we prescribe, and ultimately understand better bioavailability, toxicity, and efficacy as they relate to the composition and activity of the intestinal gut microbiome, the intestinal microbiome, excuse the oxymoron. This is not science fiction. This is published and constantly published for the last 5 or so years in Science, Nature, and other advanced journals. The leading field has been the cancer colleagues, in that they've discovered that the microbiome can influence patient response to immunotherapy. This is 3 or 4 years ago. Here's some of that data. Of course, it's not working out well, so I'll do a chop top here. But fundamentally, it's the idea that non-responder microbiota can predict that the intestinal gut microbiota at baseline can predict response to immunotherapies, in this case PD-1. I guess the only one that's legible here is, or maybe I can go through it. So the non-responder microbiota was less diverse, right? If you have patients that ended up non-responding, had a less diversity or decreased diversity in the intestinal gut microbiota. There's certain microbes that were enriched in responders, particular firmicutes, Clostridia and Clostridialis, a bunch of those that we were finding to be absent in psoriatic disease patients. And if you take a few examples, in this case, Faecalibacterium, you can see that if patients had a high abundance at baseline, they had a much higher and significantly important chance of not progressing their cancer. Okay, and then they were not happy with just the human observations. They did gnotobiotic experiments, and fundamentally what they did is they took fecal material from patients that either responded or not responded. They gavaged animals into germ-free mice. They waited for the colonization of those mice. They injected melanoma cells to promote the phenotype, and down the road they utilized the anti-PD-1 immunotherapy, and they found very elegantly that those mice that received fecal material from responders ended up having a significant decrease in the tumor size of those experimental melanoma cancer. And this is very unique. They did imaging studies, and you can see that if these animals receive non-responder fecal material, even in the presence of the immunotherapies, the tumor remained in the animal. But if those same animals receive responder material, you can see the significant decrease in the tumor in the presence of anti-PD-1, suggesting that there's something about the modulating capabilities of the intestinal microbiome that can affect systemic response to immunotherapies. And of course, we like this idea, and then we wanted precision medicine using microbiota in cancer patients to be studied in inflammatory arthritis patients, in particular responses to DMARDs, both biologic and conventional synthetic. And I'll go through this in a little bit of detail, but this is true in IBD as well. You can see that both vedolizumab, which is the anti-integrin biologic, and the response to ustekinumab significantly increases when you use microbial features in these models, right? So if you now take as an example, an AUC for the clinical response, it's about 0.6 or so. If you now add this VedoNet, which is one of the artificial intelligence networks that they use, you can see that the combination takes a much better prognostic approach, in that the AUC goes way north of 0.87 or 0.8, and that's true as well for ustekinumab in Crohn's disease, where the clinical data, again, an AUC of 0.6 or so, all the way up on the combination to 0.84. We looked at this in 17 for different reasons. The idea that IL-17 blockade may lead to a dysbiotic process, not only in the microbiome, but also in the mycobiome, meaning perturbation of the fungal homeostasis, leading to an aberrant activation of the immune system, explaining perhaps why a small proportion, and emphasis on small, of patients either develop IBD, or explaining why this 17 blockade strategy actually failed in inflammatory bowel disease altogether. We've done this, again, looking at both micro and mycobiome of patients with spondyloarthritis that received either IL-17 or TNF blockers. And we found that not only there's a perturbation in the microbial 16S community composition after the use of IL-17 compared to TNF, but also the fact that certain fungal taxa, particularly Candida, can expand in about a third of patients with spondyloarthritis that now are exposed to IL-17. And then in collaboration with Francesco Ciccia in Napoli, he looked at, actually this is his data, looked at what happens with this de novo IBD in patients that had received IL-17 blockade. And he found that unlike what happens in naturally occurring IBD, these patients post-17, actually, their biopsies were characterized by an increase in IL-25 producing TAF cells, which is curious because this is not exactly what happens in the literature of IBD, suggesting that the inflammation in the local lamina propria is, in fact, different from the naturally occurring IBD. Okay, I will finish in the last 5 or so minutes to tell you about what is it that we are doing in terms of conventional synthetic DMARDs. This is now in rheumatoid arthritis, and we'll tell you a little bit about methotrexate, which to us is an important drug. not to us, but to the field. That's what I meant. This is what happens to methotrexate. I don't know if any of you have seen methotrexate. This is that little yellowish peel. Not a big deal, but it helps millions of people worldwide. Essentially, when a patient swallows one of these or several of these, multiple things happen. The first one is that there's a high inter-individual variability of the bioavailability of the oral methotrexate, right? So the range is very wide, and it really depends on the individual. So some people may be able to absorb about 20% of the total methotrexate, and all the way up to 80% in other people. Part of it is related to the gut epithelial transporters, RFC1 and ABCC1 through 5. It doesn't really matter, but the understanding is that some of these transporters are saturable beyond 15 milligrams, and that's been demonstrated in animals at least, and there's some data in humans, although it's not perfect data set. This is known for 50 years in that methotrexate can be metabolized at least in the intestinal tract, at least in mice, right? Because if, and I'll show you the data, in a wild-type mice, this is what happens with methotrexate once you gavage animals with this drug. Most of the methotrexate ends up being biotransformed into its inactive form, which is TAMPA, right? One of the metabolites of methotrexate. And the rest, which is a minimal amount, remains as methotrexate. Now, if you treat these animals with either antibiotics or you keep them under germ-free conditions, this is what happens. There is no metabolism or biotransformation of methotrexate. Animals that do not have microbes in their intestine cannot convert methotrexate into its inactive metabolite, and therefore most of it is found in its unaltered form of pure methotrexate. Important because this is true and has been validated in other animal models. What happens in the human condition. So one is that several years ago, we asked the question as to whether or not vancomycin altered the microbiota in humans. These are new onset rheumatoid arthritis patients that were DMARD and steroid naive. And we compared to those that did not undergo vancomycin. This is an old strategy to try and decrease or eliminate one of the microbes that we were seeing in animal models. Turned out not to be true, but we learned quite a bit of things. But we looked at the gut microbiome, immunologic and clinical parameters, and what we found was that unlike what happens in vancomycin, here in red, where over time the number of operation taxonomic units, essentially species, dramatically decreased over time up to 6 months without recovering. But when you look at methotrexate over time, they didn't really change over time, suggesting that overall, the methotrexate does not have a community-altering capability in the intestinal gut microbiome. So if it didn't change the microbiome, could we utilize the pre-treatment based on gut microbiome as predictor of response in new onset rheumatoid arthritis? Just published this a couple months ago with Peter Turnbow, Renuka Nayak at UCSF, and Julia Panasson with us. And lo and behold, this is the summary slide, actually. We showed that there's a robust capability to predict drug response, in this particular case, methotrexate, in rheumatoid arthritis, mainly based on metagenomics. And I'll walk you through this. We took a new onset rheumatoid discovery cohort, and we asked the question, at 4 months, can you predict who's a responder and who will not respond to methotrexate? We did 16S, shotgun sequencing to look at the functional potential of the microbiome, and of course, 16S to look at taxonomic changes. And what we found was that indeed at baseline, there was a lower diversity on those individuals that ended up responding. Certain taxa were increased in responders. I'm not going to go into details, but these were important. But more relevant was the fact that there were significant differences in the quantity of gene orthologs that predicted response versus non-response in all these patients. We did machine learning, random forests in particular, to look at whether some of these gene orthologs or KEGG orthologs were predictive. And then we found that 38 out of 462 of those genes were very predictive of response, some with methotrexate and folate metafolate capabilities, but most of them without any relevance to the pathways, and ended up increasing the predictive value of a previously established clinical pharmacogenetic model, an AUC of 0.6, to a combined AUC of about 0.84, suggesting that not only the clinical and the genetic background of the host are relevant for methotrexate response, but also true that the microbiome may have some relevant capacity to improve response. We also did some ex vivo work where we took, based on microbiome, incubated with methotrexate for 72 or so hours, and then we measure the remnant methotrexate levels ex vivo. And what we found was curious. First, that of course there's an inter-individual variability, which we knew in animals. Now we validated in humans. And not only that, but the elimination rate of methotrexate was inversely correlated to with the future clinical response, so that the lower the elimination rate, the higher the potential clinical response of that particular patient from where the basal microbiome was derived. Here's the model. Then we think that in certain patients, certain bacterial enzymes, FPGS and others, do metabolize or biotransform methotrexate into its active form, but most of it remains intact, gets absorbed, and adds to the response downstream. When you have an increased diversity and actually quantity of these particular enzymes, now you will have more methotrexate being activated in the intestinal lumen, less methotrexate available, and therefore less absorption, which may explain why there's this variability on the downstream ACR responses and other outcomes that we see in clinical trials and in the clinic on a daily basis. But not everything is linear in medicine, particularly not in rheumatology. So I told you that this may be the case, and methotrexate does not really modify the overall community structure of the intestinal microbiota, but this is Renuka Nayak. Again, this is data that we published with them a couple months ago, suggesting that methotrexate can have off-target effects on the human gut bacteria with downstream consequence for host immunity. So the community structure doesn't change. This is the summary of this paper. What changes is the microbial pathways within those microbes. It may very well be that these methotrexate, these biotic properties, if you will, are in fact the reason for why some patients end up responding to methotrexate. More to come. There's a lot of work with humanization of mice, etc. We're doing ex vivo microbial communities and strain libraries, trying to understand the mechanisms behind this, but fascinating because it's not simple, but certainly maybe use the microbiome as determinants of drug efficacy and toxicity in inflammatory arthritis and other chronic diseases, including cancer and IBD and others. The fact that multi-omic platforms can help us generate predictive tools and help us understand the mechanism behind these changes in treatment response. And we think that we can exploit the microbiome for effective initiation of DMARDs both biologic and synthetic, conventional synthetic DMARDs as potential precision medicine strategies. And with that, I'll stop. 8:40 in the morning, just to acknowledge the people that do the work. Julia in particular, I told you about what she's been doing. Rebecca at the Psoriatic Arthritis Center, Somia as well. Rich, who has been mentored Julia in a very incredible fashion. My partner in crime for the last 10 years or so, Chris Richlin, who has been doing a lot of great work in psoriatic disease. And here we have Peter and Renuk, I told you about them, and Jose Clemente at Sinai. And I give a big shout out to NIAMS in particular. She's been instrumental in helping us fund this work. Okay, with that, I'll stop and I'll shut up. Thank you for your attention.
Thank you, Jose. That was terrific. Just to the audience, please post your questions on the question and answer tab. So we have first questions popping up. Jose, first is a note from Dr. Vinod Chandran. Congratulations, Jose, on your prestigious Henry Kunkel Award from the ACR.
Thank you. Thank you very much. The problem with that one is that it used to be the young investigator, and they took it away this year. Can you believe that? The one time I could be called young, and now gone. Anyway, thanks, Vinod. Appreciate your note.
That's great. We have a question from Dr. Jason Rockel. Very interesting data on the high fiber diet modifying cytokine expression. I was wondering if this has anything to do with changing overall circulating cholesterol levels, or if this modifies cholesterol derivatives.
Yeah, it's a great question, Jason. Thank you for that. I'm not aware of cholesterol derivatives data. What it's been looked at is some of the metabolomic alterations. And there was data at ACR by Chris Richland's group, actually, looking at bile acids. And the idea of, he has a cohort of patients that, like Daphne and Vinod have, that progress from psoriasis to PSA. And what he's done, looking at peripheral metabolomics, and what he's found on the right before progression, which is very unique, is that bile acids go down, but also butyrate goes dramatically and significantly down right before the progression into PSA. You can look at that data set at ACR Convergence. And then when people develop fully clinically evident PSA, those levels go right back up. So I don't know what I don't know that they have diet questionnaires and such, but I'm not so aware of the rest of the data. So it's curious. There are the other thing, Lichi actually has a grant to look at dietary modifications in PSA patients with Alexis Ogdi. And they are looking at the question of whether or not it has to do with anti-inflammatory properties of the diet or low calorie, decrease in weight per se. So they have two arms. They have I believe it's a Mediterranean diet on one arm, and on the other arm is weight loss by any means. And the question is, again, is it about an anti-inflammatory property of the diet, be it through high fiber or decrease in high fat diet, or is it only about decrease in total weight and BMI? We don't know. We're gonna help them with the microbiome portion of that, but that's work by Alexis and Lee.
Question, great, thank you. I've got a question, Jose, from Emmett Sandu. Very informative talk. I'm wondering if you check the effect of different levels of methotrexate on gut microbiota, particularly polyglutamate 3. Secondly, I'm thinking GGH, RFC1, ABCB1 also play an important role in metabolism, along with the FPGGS.
That's right. No, it's a great question. So here's the challenge with the polyglutamation happens in the tissue, particularly what's been done, and it's been very difficult to pursue, is in the red blood cells, right? It's hard to replicate that data. We tried via multiple ways and platforms. What we're doing, though, is we're measuring not only methotrexate, but also these new metabolites. This is an important part of our work, which is, can we understand what's the biotransformation of the methotrexate in the intestinal lumen? That's one. But the second part is, we're not going to do it in the periphery. I don't think we will be able to see metabolomic signals in the periphery for the reasons I described. But a surrogate will be the urine, because we will be able to look at the metabolites in the urine and look at the effect of the micro. And that's been done in animals. That's exactly how things are looked at, mainly because, again, peripheral methotrexate. And we've done ELISAs, we've done red blood cells, we've done circulating, just not there. It's very hard to find. But I agree with you that that's a part of the unexplained aspects of this. The other one is, parenteral methotrexate. The question always comes up, that is that the reason for why? I mean, can you bypass this conundrum of intestinal microbiome by injecting methotrexate sub-Q or IM? We don't have data. I mean, we use parenteral methotrexate, and we really don't use it as much, so we are not powered enough to do that. That's one. Two, there's this circulation, hepatobiliary circulation of methotrexate that we believe will be subjected to the same transformation in the intestine as it circulates through the liver and beyond.
Next question is by Vinod Chandran. Any attempts in understanding the pharmacomicrobiology of oral versus subcutaneous methotrexate?
I think I just anticipated that question from Vinod.
Exactly.
If you have patients, send them along, because we're very intrigued. Patients on sub-Q. What happens in the United States, as you know, is that someone came up with the idea that they can patent sub-Q methotrexate and then take the $50 a month price tag to $5,000. And so here we are, so we really use, and then we have shortages of methotrexate, mainly because they use it for chemotherapy and because it's so inexpensive that people don't want to have they don't go through the trouble of generic methotrexate anymore. We're about to buy methotrexate from Canada, by the way. So if you want to create some business, I'm up for it.
Question, Jose from Kelsey Collins. Fantastic talk. What is known about the potential role of the microbiome in patients who become resistant to previously successful therapies?
It's a great question. I don't think we know much about that. It's a wonderful question. Are you saying secondary
failure?
Secondary failure. That's an intriguing one. I mean, I can think about methotrexate again based on the data published on cell host microbe, in that it may very well be that you continue to expose these patients to these off-target effects of methotrexate, and at some point you modify the microbiota in such a way that now you converted the microbiome into a non-responder microbiome. That's part of what we're trying to discern right now. Now, is that true for biologic DMARDs? I don't know. That's much more complex, right? I think with the immunotherapy story, it's intriguing. I don't know. It's hard to connect the dots, right? The idea for the biologic systemic therapies is that you prime the immune system through your intestinal microbiome in order to respond better to the drug. That's the hypothesis. And so if the idea is that you're going to stop responding to a TNF blocker, for example, to which a patient responded for, say, 5 years, and that's because of the perturbations we see, and I showed you some data on TNF and 17, that's possible. It's a leap of faith, though. I don't know that it's that robust. Needs to be studied for sure.
I would say just to comment on the anti-IL-17 data, which was interesting, in contrast to what the anti-IL-23 story is. You've got the strange dynamic in axial SpA, so the 23 blockers work in IBD, but not in AS. The 17 blockers, exactly the opposite. So do we have any data on the anti-IL-23 effect on the microbiome?
Yeah, actually, there are 2 or 3 pieces of data. One is from Ranjani Thomas. She has data on the SKG model, actually. And I think what the slide that didn't work well was from her study, in that IL-23 does not really perturb the microbiota in a, say, dysbiotic manner. And then there's data on humans. I showed you data on ustekinumab. They show that there's not that much, that many changes over time at all. The idea of IL-23 being an immunomodulator more than an immune suppressant pathway, right? So that may very well be true. It's such a pleiotropic effect, right? It comes all the way up from dendritic cells, antigen presenting cells. So you might expect it to be more of a peacemaker, IL-23, than other downstream molecules such as TNF and 17, for sure.
There's a follow-up question by Vinod. Weight loss often leads to reversing secondary failures. So likely, is it not?
Reversing secondary failure, so likely, is it not? Yes. Yes. There's data from the Italian group, and Vinod, you know this data better than I do, but yes. I guess this is a comment more than a question, right?
I think it's just related to the previous comment and the question around methotrexate.
Yeah. I don't know the answer. There's data on, actually, the entire field of the microbiome came to age in work comparing monozygotic twins discordant for obesity and leanness, if that's a word. And the finding is that if you lose weight, you not only modify the microbiota, but you modify the metagenomics, meaning what kind of functionality the microbiota has, and now you reset your microbiome through diet. So I don't know the answer to this, but it may very well be true that it affects the way we metabolize medications in that manner. And it may be important for psoriatic disease more than it is inflammatory, for example, being that psoriatic patients tend to have higher BMI, etc.
I would say we have a number of patients in my clinic asking about probiotics.
Yeah.
So yeah, I guess they find that more acceptable than FMT. I don't know. Anything encouraging on the probiotic side?
So there's some data actually published on some probiotics, particularly in IBD. There have been clinical trials actually showing some benefit. Overall, the problem with probiotics is that we don't know what they have. Number one, right? If you go to the supermarket or whatever the equivalent in Canada for CVS or Walgreens, or the yogurts, whatever, those are, and it's been studied. I mean, if you look at those lactobacilli, they just don't survive a pH of 1 in the stomach, right? So, reconstitute your flora, but yes, guess what? It just died in your stomach. So that's one. But two, is that I think the field is moving away from probiotics, and it's mostly about prebiotics, short chain fatty acids being one such example. And there's work being done. We have one study ongoing. It's not to improve disease outcomes, but mostly to modify disease methotrexate response. We're trying to take our findings and try and modify the non-responder microbiota at baseline into a responder as much as we can make that happen. And so that may be one aspect of this. Can you now improve response just by incorporating short-chain fatty acids, for example.
And as we move to oral biologics with the JAK inhibitors, is there anything, I mean, can we suspect that the gut microbiome might alter absorption of oral JAK inhibitors?
That's a great question. So there's data from a year or two ago, published in Science by the Andy Goodman's group. What they've done is that they took a big platform, metabolomic platform, and they asked the question of these, I think it was 300 common drugs, how many of them are being metabolized by the gut microbiome? These are not antibiotics, right? These are the thiazide, there's like, calcium channel blockers, there's like, antifungals, antibiotics, etc. But the vast majority of them, common drugs, about 2/3 or more of those drugs are modified by the gut microbiome, which now opens this Pandora's box of what do we do with all of this? It was true for sulfasalazine, but it was rationally designed to be cleaved by the gut microbiome. But if it's also true for beta blockers and calcium channel blockers, then we have a problem or a potential opportunity.
Right.
And so this is just the beginning, in many ways. The AMP consortium will have a big microbiome presence just to answer those questions.
Right.
And hopefully we can contribute in other diseases, right? The question of hydroxychloroquine in lupus. I mean, we don't even know much about it. A drug that has, anti-malarial capabilities, assuming it has antibiotic properties as well. We don't know that. So JAK inhibition will be part of this.
Sure. Right.
One last question. If we were going to fast forward to the next few years, and it probably relates to other omics strategies as well, how do you see the future of microbiomics fitting in the real-time clinical care decision making?
Right. One hope, and again, there's like everything else. If you remember, the Human Genome Project was gonna cure every disease in the planet. We're gonna sequence everybody's genome, and that's it. Guess what? It just made a lot of money for 23andMe and all those people, and we probably found, genetic etiology for about 1 or 2% of our diseases. So there's going to be a lot of disappointment. I'm going to start with that, because this is not the ultimate. This is part of multi-omics, as you very well described this. I think that we will have to learn how to be precise with this. I guess the prebiotic story is one such approach, where the idea will be to modify the bioreactor that is the intestinal microbiome to help with the already available therapeutics. That to me will pan out and will be helpful down the road. In terms of biomarkers, we can see this happening in the clinic, right? If you're able to do this in a low throughput fashion, in an inexpensive fashion, say, you can do a qPCR on some of these microbes that with a turnaround time of a day for $5. I mean, you can discriminate who may want to skip methotrexate, for example. That's one idea there. FMT is going to be difficult. That's not practical. Who's going to do this? Certainly a lot of people are asking for that. I don't know that it's practical. The question is, can you modify? And then prediction is important. Prediction of disease and prediction of disease outcomes therapeutically. But just like everything else, it's medicine, right? We're going to fail more than we're going to succeed. Just the 5% that we're going to succeed on, we hope will contribute in the next 5 to 10 years. For sure.
Great. We're right on time. It's 9 a.m. here. Thank you, Jose, for joining us today. Terrific lecture and great discussion.
Thank you, Jose. It was wonderful. Thank you very much.
Thank you, everybody, and hope I can make it to Toronto soon. I love Toronto.
Absolutely. Thank you. And thank you to the audience for joining us today. See you all on December 14th. Next lecture is by Dr. Lee Callahan from University of North Carolina on social determinants and arthritis. Thank you, everyone. Thank you, Jose. See you all. Bye.
Bye. See you.
Thank you so much, guys. Have a good day.
Take care. Have a good day.